So, I want you to think about what might just be the scariest word in all of medicine.
Yeah, it's a word that, instantly changes the temperature in a doctor's office.
Exactly. Metastasis. We all know the word. Like, if you hear it, you immediately know it means the cancer has spread.
Right.
But based on the massive stack of sources we are diving into today, and I mean a stack that spans everything from handwritten autopsy records from the 1800s to modern genomic sequencing and live animal imaging, almost everything the general public believes about how this process actually works is slightly or sometimes wildly wrong.
It is entirely misunderstood, honestly. When we talk about cancer or, when you read headlines about cancer, the public imagination usually goes straight to the primary tumor.
Right, like the original lump.
Yeah, exactly. We think about shrinking it or cutting it out, radiating it. But understanding the exact pathophysiology of how cancer actually spreads, the literal mechanical steps of how a cell travels from one organ and colonizes completely new tissue, that's the entire focus of modern oncology.
Wow.
Yeah, we really have to look at the deeply intricate biology of the spread itself, not just the original tumor.
And the stakes here, I mean, they could not be higher for anyone listening. Because according to this highly influential 2011 review by Chafffer and Weinberg in the journal Science, metastasis accounts for roughly 90% of all cancer deaths.
90%. It's staggering.
90%. So our mission for this deep dive is to figure out exactly how these cells manage to pull this off.
Okay, let's unpack this. If a primary tumor is like a localized rebellion, metastasis is a coordinated global invasion. But how do they actually execute that invasion?
Well, before we can even track that invasion, we have to clear up a massive identity crisis about what a metastatic cell actually is.
Right.
We need to start with the fundamental difference between a benign growth and a malignant one. People often think a benign tumor is just, a small one.
Right, it's just not big enough to be a problem yet.
Yeah.
Exactly. But it has absolutely nothing to do with size. A benign tumor, no matter how massive it gets, stays put. It completely lacks the biological machinery to leave its neighborhood.
Oh, I see.
A malignant tumor, on the other hand, possesses the capacity to metastasize. Even if it never actually does in a specific patient, it possesses the cellular tools to do so.
Which leads to what seems to be the biggest clinical misconception out there, the naming convention. Like say you have a loved one who had breast cancer, and years later, the doctor finds tumors in their liver.
Yeah, it's a terrifying scenario.
The immediate heartbreaking instinct is to say, oh no, now they have liver cancer. But according to these sources, they don't.
No, they absolutely do not have liver cancer. They have metastatic breast cancer or breast cancer with liver metastasis.
Okay.
This is where the concept of pathophysiology really matters. If a pathologist takes a biopsy of that tumor in the liver and puts it under a microscope, they aren't looking at distorted liver cells.
Wait, really?
No, they are looking at tissue that is histologically and genetically breast tissue. It is literally a piece of the breast growing inside the wrong organ.
Wait, if the cells are sitting in the liver, surrounded by liver cells, bathing in liver blood, why don't they just adapt and act like liver cancer? Are they wearing some kind of, biological disguise to hide from the liver?
What's fascinating here is that they don't need a disguise because they retain their original genetic identity.
Really?
Yeah, they bring their own architectural blueprint with them. And this isn't just medical pedantry, It has massive life or death treatment implications for anyone walking into a clinic.
Because it changes the drugs they use.
Exactly. Because those cells retain the genetic identity of the original breast tumor, they also retain its specific treatment vulnerabilities. That is why oncologists will treat that tumor in the liver using breast cancer drugs, not liver cancer drugs.
That is wild to think about. It's a breast cancer cell setting up an entirely foreign colony in the liver. But because these cells keep their original identity and don't just blend in, it begs a massive historical question.
Right.
How do these wandering cells decide where to set up camp in the first place? Like, do they just get off the highway at a random exit?
That exact question has driven a century-long debate in medicine. The word itself, metastasis, comes from the Greek methystani, which means to change or to remove from one place to another.
Interesting.
Yeah, a French surgeon named Recamier actually introduced the term into the medical literature way back in 1829.
Wow, that early.
Yes, but the real debate about how cells choose their destination kicked off in 1889 with an English surgeon named Stephen Paget.
Oh, yeah, I saw Paget's name all over the research.
Right.
What was he doing in 1889 to figure this out, especially before modern genetics even existed?
Well, Paget took this brilliantly simple mathematical approach. He looked at autopsy records from 735 women who had died of breast cancer. He reasoned that if cancer cells just spread randomly, wherever the blood carried them, they should end up evenly distributed across the body based purely on how much blood an organ receives.
That makes logical sense.
Right, but the math didn't add up. The breast cancer showed up in the liver, the lungs, and the bones all the time. But it almost never showed up in the spleen.
Even though the spleen filters an enormous amount of blood.
Exactly.
The cancer cells had to be reaching the spleen, right? I mean, the blood is taking them there.
Exactly. They were arriving at the spleen, but they just weren't growing there. This led Paget to propose the famous seed and soil hypothesis.
Seed and soil?
Yeah. He argued that the tumor cells are the seeds, and they can only take root and grow in organs that provide hospitable soil.
Oh, I see.
So if a breast cancer seed lands on the hostile, concrete-like environment of the spleen, it simply dies. But if it lands in the fertile, nutrient-rich soil of the bone marrow, it thrives.
That makes intuitive sense. You can't plant tomato in the desert, right? But looking at the historical timeline, the medical community didn't just accept this and move on, did they?
No, they definitely didn't. A few decades later, in 1928, a very prominent pathologist named James Ewing argued that Paget was overthinking the biology.
Okay, so what did he think?
Ewing proposed a purely mechanical, anatomical explanation. He basically said it was just plumbing.
Plumbing.
Yeah. For example, he pointed out that colorectal cancer spreads to the liver not because the liver is some magical soil, but simply because the blood from the colon drains directly into the liver via the portal vein.
Oh, so it's literally just the first stop on the anatomical train line.
Exactly. And Ewing's mechanical plumbing theory actually dominated for decades, completely pushing the seed and soil idea into the shadows.
So, which one is it? Are these cancer cells just floating down a river until they randomly get stuck in the plumbing? Or are they like extremely picky house hunters looking for a very specific neighborhood with good schools?
Today we know it is actually a synthesis of both. The anatomy, the physical plumbing, dictates where the cells physically arrive first. But the biology, the soil, dictates whether they can actually survive and colonize once they get there.
Right.
And the definitive biological proof for Paget's soil idea didn't come until the 1970s, thanks to a researcher named Isaiah Fidler.
What did he do?
He ran some groundbreaking experiments using a specific mouse melanoma model.
Wait, how do you prove that a cell is picky about its soil using a mouse?
Well, Fidler isolated cells from a metastasis in one mouse and injected them into the bloodstream of a new mouse.
Okay.
He found that those cells produce far more metastases than cells taken from the original primary tumor. He basically proved that the ability to spread isn't just a random accident that any cancer cell can stumble into.
Wow, so it's specific.
Highly specific. It's a heritable trait possessed by specific subclones of the tumor, and they pass that trait on. This gives us the modern clinical concept of organotropism.
Organotropism, meaning a preference for a specific organ.
Precisely. It explains the highly predictable destinations for certain cancers. Prostate cancer strongly favors the bone.
Right.
Uveal melanoma, which is a cancer of the eye, almost always goes to the liver if it spreads. They are specifically looking for their soil.
Okay, so if spreading requires this intense combination of the right anatomical plumbing and the perfect biological soil, what does that actual journey look like for a single microscopic cell?
It's intense.
Because reading through the pathophysiology in these sources, it sounds less like a journey and more like an absolutely lethal obstacle course.
It really is a gauntlet.
Yeah.
Biologists call it the invasion metastasis cascade, and it happens in 5 brutal steps. A cell has to succeed at every single one in order or it dies.
Okay, what's step 1?
Step 1 is local invasion. The cells at the edge of the primary tumor undergo a process called EMT, or epithelial-mesenchymal transition.
Okay, I need an explanation for that because epithelial-mesenchymal transition sounds like pure medical jargon. What is the cell actually doing?
Fair enough. Let's break it down. Epithelial cells are the cells that line the surfaces of your body and your organs. Normally, they're tightly glued to their neighbors to form a solid barrier.
Right.
During EMT, the cancer cell basically hacks into a genetic program from normal embryonic development. It turns off the genes that make the biological glue, specifically a protein called E-cadherin.
So it loses its grip.
Exactly. It loses its regular shape, becomes highly mobile, and starts secreting these enzymes called MMPs, matrix metalloproteinators.
So it unglues itself from the tumor. But what do those enzymes, the MMPs, do?
Think of MMPs as biochemical scissors. The newly mobile cell uses these enzymes to literally chew through the surrounding structural proteins and tissue boundaries. It dissolves the neighborhood so it can break out.
It's chewing its way out of the neighborhood. Okay, but to go global, it needs a highway. How does it actually get into the bloodstream without rupturing the vessel?
That is step 2, extravasation. The mobile cell navigates toward a nearby blood or lymphatic vessel, which, by the way, the tumor has often tricked the body into growing.
Ugh, sneaky.
Very. And then it pushes its way through the vessel wall into circulation. It essentially squeezes between the cells lining the blood vessel. Now, it is officially a CTC, a circulating tumor cell.
And once it's in the bloodstream, it's on a fast track to anywhere in the body.
Right.
But the sources point out that this is actually the most dangerous part of the journey. If the blood is basically just a transit highway, why is it so deadly for these cells? Has anyone actually watched this happen?
We have, actually. In 2002, landmark imaging studies tracked these individual cells in living animals, and they found that well under 1% of the cells that enter the bloodstream actually survive to form a secondary growth.
Under 1%.
Yes. The bloodstream is incredibly hostile. First, the physical sheer stress of the blood pumping can literally tear the cell apart.
Wow.
Second, circulating immune cells are actively hunting them. But the most fascinating hurdle is something called anoikis.
Anoikis. It's a Greek word for homelessness, isn't it?
It is, yeah, and it describes a form of programmed cell death. Normal cells are hardwired with a safety mechanism. If they lose physical attachment to their cellular matrix, they self-destruct.
Oh, to keep them in line.
Exactly. This is designed to prevent rogue cells from just floating away and growing wherever they want. For a cancer cell to survive in transit, it has to completely override this existential suicide program while floating freely in the blood.
Here's where it gets really interesting to me. If well under 1% survive the sheer stress, the immune attacks, and the self-destruct programming, this is a spectacularly inefficient process.
It really is.
Why is metastasis so deadly if the cancer is fundamentally so bad at it? Is it just a numbers game?
It is a terrifying numbers game, and arguably the most counterintuitive fact about cancer. The process we fear the most is biologically highly inefficient for any individual cell.
Right?
But a primary tumor the size of a golf ball can shed millions of cells into the bloodstream continuously, every single day. The cancer just overwhelms the abysmal odds through sheer volume.
Millions of cells just rolling the dice constantly. So what happens to that tiny fraction of a percent that actually survives the river ride?
They reach step 4, arrest and extravasation. The surviving cell gets physically trapped in a narrow capillary bed in a distant organ, often simply because the cancer cell is physically too large to squeeze through the tiny blood vessels.
It just gets stuck.
Yeah. And once it is wedged in there, it uses those same biochemical scissors from step 1 to push its way back out of the blood vessel and into the new organ tissue. Which brings us to the final hurdle, step 5, colonization.
This is where Paget's soil concept finally comes into play. The seed has landed.
Right, but landing isn't enough. The cell has to survive in a totally foreign microenvironment. It has to hide from the local immune system in the liver or the lungs.
Okay.
And crucially, it often has to trigger a mesenchymal epithelial transition. It reverses step 1.
Oh, so it glues itself back together.
Exactly. It gives up its mobility, reestablishes cell adhesion, and settles down to grow into a solid mass. But here's the most sinister part. Many cells that successfully reach this stage don't grow right away. They enter a state of dormancy. They go to sleep, sometimes for decades.
Wait, so a cell survives this impossible evolutionary gauntlet, gets into the bone marrow, and then just camps out totally undetected, doing absolutely nothing for 20 years.
Yes, but recent science shows the primary tumor might actually be helping them from afar before they even get there.
Wow.
In 2005, a major paper was published by David Leiden showing the existence of a pre-metastatic niche. Before the tumor cells ever leave the primary site, the tumor sends out chemical signals, including specific bone marrow-derived cells called VEGFR1 positive cells.
What do those cells do? Because that sounds like alphabet soup.
Think of VEGFR1 positive cells as biological construction workers. The primary tumor dispatches them into the bloodstream. They travel ahead to the target organ, say the lungs, and they actually start laying down new blood vessels and suppressing the local immune system before the cancer cell even arrives.
What, kidding?
No, the tumor actively prepares the landing zone, fertilizing the soil in advance.
That is incredible and completely terrifying. The tumor is sending out advanced scouts to build a safe house.
It's exactly that.
But if these cells can prepare the soil early and then lie dormant for decades, that completely upends how we traditionally think about the timeline of cancer. Like, I always thought you get a lump, it gets bigger, and then if you wait too long, it spreads.
You just described the linear progression model. And for most of the 20th century, the medical establishment believed exactly what you just said.
Really?
Yeah. The idea was that cancer grows locally, gets big, invades nearby tissue, and only at the very absolute end of its life cycle does it shed cells that spread. A strictly ordered late-stage event.
If they thought it just grew outward in a straight line, why didn't surgeons just cut wider and wider around the tumor to make sure they got it all?
Well, they tried exactly that. That linear belief drove William Halsted's surgical philosophy in the late 19th century, the radical mastectomy.
Oh, wow.
Halsted believed that breast cancer spread in an orderly, contiguous line down the lymphatic system. So his solution was to remove the breast, the underlying chest muscle, and all the axillary lymph nodes in one massive disfiguring block. He believed if you just cut wide enough, you could outrun the physical spread.
But it didn't work. The cancer still came back in the bones or the lungs.
It failed because the biological model was wrong. It took decades, but large randomized trials in the 1970s, led by Bernard Fisher, finally proved that radical mastectomies offered no survival advantage over much less disfiguring surgeries.
That must have been a huge shift.
Huge. Fisher realized that cancer often spreads systemically through the bloodstream well before we can even detect the primary tumor.
So, what does this all mean for our timeline? We used to think of cancer like a train making scheduled sequential stops, but this new theory, the sources mentioned, the parallel progression model, means it's more like a network of sleeper agents.
That is the perfect analogy. Genomic sequencing now shows that cells can disseminate when the primary tumor is microscopic, perhaps only a few millimeters across.
Wow.
Those disseminated sleeper agents then evolve independently at the distant site in parallel with the primary tumor. And this raises an important question. If these cells disseminate so incredibly early, is catching cancer early through screening as fully protective as we've always assumed?
Because even if you find a primary tumor when it's tiny, the sleeper agents might already be hiding in the bone marrow.
Exactly.
I know there was a lot of hope placed on finding the genetics behind this. The sources talk about the discovery of metastasis suppressor genes like NM23 in the late 1980s. They found the genes that stop cancer from spreading. But it was a massive failure clinically. Why couldn't we just turn those genes into a drug?
Because of that exact timeline problem. By the time a patient is sitting in a clinic and we detect that they have a visible tumor, the dissemination has often already happened.
It's too late.
Yeah, the disease is already systemic. You can't give a drug to prevent the horse from leaving the barn if the horse has been gone for 5 years.
That makes painful sense.
That is why we have almost no drugs on the market that specifically restore metastasis suppressor pathways. We have amazing drugs that shrink tumors, but preventing the mechanical spread after the fact is incredibly difficult.
Which brings us to what this means for you, the listener, if you or a loved one are navigating this today. First of all, we should clarify, metastasis is not universally a death sentence, is it?
It is not. The prognosis changes dramatically, of course, but certain metastatic cancers, like metastatic testicular cancer and some lymphomas, remain highly curable even after they have spread widely.
Oh, that's good to hear. Why is that?
This is simply because those specific cells remain exceptionally sensitive to systemic chemotherapy, no matter where they are in the body.
But for the cancers where it is highly lethal, this intricate biology dictates exactly how doctors treat you. The sources talk about liquid biopsies.
Yes, those are revolutionary.
Because we know cells are shedding into the blood, instead of surgically cutting into a lung tumor to see if it's mutating, doctors can just take a standard blood draw and actually count the CTCs, the circulating tumor cells, to see if the cancer is active or responding to treatment.
Yes, and this biology dictates imaging too. Because of organotropism, if a patient is diagnosed with prostate cancer, the oncologist isn't going to waste time ordering a scan of their spleen or their brain.
Right, because of the soil.
Exactly. They're going to order a bone scan because they know exactly which soil that specific seed prefers.
Here's something I think a lot of us wonder, especially if you know someone who has been through treatment. If someone has their visible tumor completely removed by surgery, the surgeon comes out and says, "Great news, we got it all." Why do they still have to sit through months or years of chemotherapy? Are
Yeah.
doctors just blasting the whole body just in case?
Well, if we connect this back to the bigger picture of tumor dormancy,
Yeah.
makes perfect sense. The surgeon absolutely got all the visible cancer. But a scalpel cannot cut out microscopic sleeper agents hiding in the bone marrow or the lungs. The adjuvant chemotherapy, or the years of maintenance therapy patients endure, is specifically designed to target those invisible, disseminated cells.
So they're going after the sleeper agents.
Exactly. We are trying to aggressively maintain the dormancy or kill the stragglers so the disease never wakes up and colonizes.
It is a constant holding pattern. Okay, let's step back and summarize this incredible, terrifying journey.
Let's do it.
The scariest word in medicine is actually the result of a spectacularly inefficient biological process. A primary tumor sheds millions of cells. They use biochemical scissors to unglue themselves and chew through tissue.
Right.
They enter the bloodstream where almost all of them die from sheer stress, immune attacks, or their own internal self-destruct programming. But a tiny fraction survive, get wedged in a distant capillary, and push their way into new tissue that the primary tumor may have already sent biological construction workers to prepare. And once there, they might just go to sleep for a decade.
It is an evolutionary gauntlet, and only the most resilient, adaptable clones survive it.
Wait, thinking about those dormant cells and the fact that they evolve independently in parallel, imagine a patient has a primary tumor removed.
Okay.
The surgeon says they're clean, but one single cell escaped early and went dormant in the bone marrow. For 10 years, that dormant cell has been sitting there, facing different pressures, evolving completely on its own.
Yes.
A decade later, it wakes up. If we treat that patient today based on the genetic profile of the primary tumor we threw in the incinerator 10 years ago, aren't we fighting a ghost? Like, are we accidentally applying selective pressure that forces that sleeper cell to become an even more resilient, untreatable superclone because we are using the wrong weapons?
That is the bleeding edge frontier of oncology right there.
Mm.
The dormant cell is not the same cell we cut out 10 years ago.
Wow.
It is why treating metastatic disease is the ultimate moving target, and why understanding this pathophysiology is the most urgent challenge in medicine today.
Definitely something to mull over the next time you hear a headline about a miracle cure for cancer. We want to thank you for joining us on this deep dive into the hidden microscopic world of metastasis. Keep questioning the world around you, and we'll see you next time.